Hey guys, welcome back to Rayyann Alqadry. Over the course of these few months, I realized that not many people knew what COVID-19 actually is. So I decided to create this video series explaining exactly what COVID-19 is. All the information collected in this video series were from the internet, and they were interpreted by a few individuals, none of who are virologists or microbiologists. So any discrepancies in the interpretation will be caused by the little exposure we have in virology and microbiology. And for today's episode, we would like to compare two very different things, namely coronavirus and COVID-19. Now, to understand why coronavirus and COVID-19 are two different things, we have to first get a general idea of what coronaviruses themselves are. Now, coronaviruses are any virus in the family of Coronavidae. That is Coronavidae, Coronavidae. That's a family of all these coronaviruses. Now, coronaviruses, the average coronavirus has the incubation period of about 3 to 4 days, which means that once you're infected with a coronavirus, you usually have 3 to 4 days of no symptoms till you actually get symptoms. Now, what are the symptoms? Well, you've got things like your fever, coughing. These are all symptoms of the common cold. As a matter of fact, 10 to 15% of all common cold cases are from the coronavirus. Now that we've gotten an idea of what coronaviruses are, let's move on to classifications. Now that we have a general understanding of what coronaviruses are, we can look into the classifications of coronaviruses. Now, coronaviruses fall under 4 main classifications. You've got alpha coronaviruses, beta coronaviruses, delta coronaviruses, and gamma coronaviruses. Now, there are 7, according to CDC, there are 7 human coronaviruses, which means any coronavirus that has infected a human being. What are those 7? Well, you've got the 22NE, an alpha coronavirus, together with the NL63. They are both alpha coronaviruses. Then you've got the OC43 and the HKU1. Both of them are beta coronaviruses. Now, besides these 4, the other 3 include the SARS, or the Severe Acute Respiratory Syndrome coronavirus, the MERS, or the Middle East Respiratory Syndrome coronavirus. Then you've got the novel coronavirus of 2019, the Severe Acute Respiratory Syndrome coronavirus number 2, or simply SARS-CoV-2. Now that we've got an understanding of the classifications of coronaviruses, we can look at the structure of coronaviruses, which is actually what gives it its name. Now, before we look into the structure of coronaviruses, we should get a good understanding of the makeup of DNA. So what do I mean? DNA, when you break it down, it's sort of like a double staircase, a double spiral staircase. When you break it down, you get single spiral staircases. These single spiral staircases are what we call RNA. So when you break DNA down, you get simpler strands of what you call RNA. DNA is deoxyribonucleic acid. RNA, you remove the deoxy part and you get ribonucleic acid. So these RNA, they comprise of even smaller things called proteins. These proteins are actually just chains of these other smaller things called amino acids. Now, amino acids, once you break them down, you get to the most basic form, you get elements. So elements such as oxygen, elements such as hydrogen, elements such as nitrogen, things that we all require to live. So to recap, from your elements, you put them together, you get amino acids. Amino acids in a chain create proteins. Proteins put together create RNA and DNA. Now, with that out of the way, we can analyze the structure of a coronavirus. Now, according to NCBI, coronaviruses are spherical or pleomorphic envelope particles containing single-stranded positive sense RNA associated with a nucleoprotein within a capsid comprised of matrix proteins. the envelope bears club-shaped glycoprotein projections. Now, it is a mouthful, but if we remove the pleomorphic and the nucleoprotein part, it's actually much easier to understand. Starting from the first part, the spherical envelope particle, all that means, you see, an envelope is an outer layer of a virus. It is the outermost shell of the virus. It is what covers the virus. And the spherical part, all it means is that it is a sphere. Particle is the term used because of how small it is. So now we know what spherical envelope particle is. So it's just this very small spherical virus. Okay. Then we've got the single-strand positive sense RNA. Now we know what the RNA is. The most important part of this is the positive sense part. What does positive sense mean? Well, in virology, sense refers to whether this strand of RNA can be what we call a messenger RNA. Now, what is this messenger RNA? You see, our body is made up of these small things called cells. In those cells, there are sort of workmen. These workmen are what we call ribosomes. Now imagine that these ribosomes can only speak in one language. They can only communicate, they can only read in one language. Messenger RNAs, you see, RNAs can be written in many different languages. A positive sense RNA is one written in the language that the ribosomes can understand, that they can read. Why is that important? Because what the virus does is that once it enters the body body itself will start manufacturing more of those viruses. And in order to do that, it needs to pass the instruction to these workmen. So because it is a positive sense RNA, they don't need to go through any process to make it readable by these ribosomes. So the ribosomes can take in the instructions and they can begin the process of multiplying and creating new viruses. So that's what that positive sense part means. Because there are negative sense viruses, those will have to employ a different technique in order to change their instructions to a language that's readable by the ribosomes. So we understand what positive sense is. Now we've got the capsid. The capsid comprised of matrix proteins. First, let's look at the capsid. This capsid is sort of like, it's an inner shell inside that outer shell. So you've got the outer layer, then you've got inside that another smaller shell where the RNA actually is. Now this shell, it's made up of, as they said, matrix proteins. Matrix proteins are just, think of them as proteins put together in a network. Sort of like that, the blockade scene in Guardians of the Galaxy. The last part in our understanding of the structure of a coronavirus is the part that it talks about this club-shaped glycoprotein projections. So first of all, I'd like to mention that this is what makes it called coronavirus. You see, we know what the virus part means, but the corona is Latin for crown. And these crowns, these are the shapes that you find around that spherical virus. Okay, so these spikes out there, they're called, they are spike proteins, and they create what they call club-shaped glycoproteins. So what are the glycoproteins? Glycoproteins, they're just proteins, as we talked about before, proteins with carbohydrates attached to them. So small, tiny particles of carbohydrate that are attached to the actual protein itself. So altogether, this entire thing creates the structure of the coronavirus. Now, why does it need these spikes? To understand that, we're going to need to look at viral entry. In virology, viruses have got three ways of entering a cell. The first is by something called membrane fusion. Now, how membrane fusion works is that the envelope of the virus and the outer layer or the outer membrane of the cell will merge together. You see, there's fusion and there's fission. Fission is when you break it apart. Fusion is when they go together. So as they go together, whatever is inside them will join together. So whatever is inside the virus will enter the cell itself. Now the second way is by something called endocytosis. Endocytosis is usually done by viruses with no envelope. So if they just have the capsid will push on the outer membrane of the cell, and eventually part of the membrane will warp around it, creating what you call a vesicle. The vesicle will then transport it through the cell, where it will eventually break off, and the genes are there for the ribosomes to replicate. Now the third way is by something called genetic injection. Genetic injection is usually happens when you've got receptors around the cell and parts of the virus attached to those receptors. What will happen is that the genes are then injected into the cell, and the outer layer, that envelope, it just stays on the surface. Now coronaviruses, some coronaviruses employ the membrane fusion techniques. Some coronaviruses like the MHV, the murine coronavirus, which attacks mouses, they employ the membrane fusion method. However, some like SARS, the original SARS, and this COVID-19, they employ the endocytosis method of viral entry. But whatever the method is, they will always have receptors, and these receptors are necessary for all viral entry. Whether it's membrane fusion, in membrane fusion, the receptors are what causes the membrane to start fusing in together. In endocytosis, it is the receptors that creates the warp and allows the membrane to break off, creating the vesicle. It's also receptors that allow the genes from viruses to be injected in. Now, in coronaviruses, there are many receptors that are used. In fact, according to NCBI, that is one of the most outstanding features of coronaviruses, the fact that there are so many different coronaviruses and they use so many different receptors. For example, the NL63 coronavirus, that goes to a receptor called the APN receptor. The MERS virus, it goes to a receptor called the DPP4. And viruses like the OC43, that binds together with what we call acidic sugar, which brings us to COVID-19. The virus that's responsible for COVID-19, which is called the SARS coronavirus number 2, or SARS-CoV-2, its receptor is one by the name of ACE2. Now that we've been brought to COVID-19, we can start comparing some subtle differences between coronaviruses, the average coronavirus, and COVID-19 and the SARS coronavirus number 2. Two of the main differences are the symptoms and the incubation period. So symptoms, in terms of symptoms, we all know that it's more severe. Its symptoms include sore throat, they include diarrhea, they include a lot of other more deadly things that will eventually, if not taken care of, lead to one's death. Now, when it comes to the incubation period, according to the WHO, the incubation period is about 1 to 14 days. So after 14 days, if you don't have any symptoms, you should not have COVID. You should not have COVID-19. However, there have been cases where incubation periods last 19, 24, and even 27 days. So the exact incubation period has not exactly been determined, because for all we know, if we say that it is 30 days, one case may come up with an incubation period of 40 days. So research has not been completely comprehensive when it comes to the incubation periods of this SARS coronavirus number 2. Now, one final question. Why is it so deadly? Why is this SARS coronavirus number 2 so deadly? Ladies and gentlemen, that question is one that will be answered in our next episode of What is COVID-19? Goodbye.